Mark, you can't try to do that voice. All right, darn. Okay, well, even without that, I'm super excited to be here. I think, like all of you, it's just great to get the gang back together, so to speak, in terms of OIS and this great conference. So great to be here. Great to have the chance to give you an update on where we're at REGENXBIO. We are a publicly traded company, so here's our forward-looking statements. So REGENXBIO, we are a company that has a mission to improve the lives of patients through the curative potential of gene therapy. And the way we have gone about this over the last over 10 years has been leveraging the special and unique characteristics of the NAV platform of AAV vectors, particularly the ones listed here. And I'll particularly call out AAV8, which we use in our lead program, RGX-314, and also AAV9, where this technology has enabled the advancement of even commercial programs such as Zolgensma for the treatment of spinal muscular atrophy, and many other clinical stage programs, both in the eye and outside the eye, with us and with our partners. Here you see our internal pipeline, which includes a lot of very exciting non-ocular, mainly monogenic disorder-based treatments, including in CNS, and also soon to start for Duchenne muscular dystrophy. But of course, today I'm going to focus on the eye and on our lead program, RGX-314. And we've had a lot of significant milestones recently. One I'll start off with, which we announced a few weeks ago, is our strategic partnership with AbbVie for the development and commercialization of RGX-314 for wet AMD, DR, and potentially other indications. We're very excited about this collaboration and this partnership, and really the complementary skill sets that we can bring together to really advance RGX-314 development globally. So now I'm going to step back and talk about RGX-314 for the treatment of wet AMD and DR. So I'll start with the broad message without details, because I think this whole room knows this, because I think many of us have worked in this space and recognize the significant unmet need that exists for these VEGF-driven retinopathies, where we clearly know that repeated anti-VEGF injections into the eye work for these indications. And we see that over and over again in well-controlled studies. But unfortunately, we also see over and over in real-world follow-up studies and even follow-up studies from these well-controlled studies that over time, patients are simply not getting the anti-VEGF injections they need. So we're very interested in applying one-time gene therapy as an approach that really could address this unmet need. So RGX-314 specifically utilizes the AAV8 vector, which is hypothesized to have greater and longer duration of protein expression, particularly in tissues that it targets well, such as the retinal tissue itself, and also to have lower immune response compared to earlier generation AAV vectors. And the vector itself contains a transgene for an anti-VEGF monoclonal antibody fragment, with the goal to have the patient's own retina cells produce in a sustained fashion the anti-VEGF protein. This slide shows the 4 studies that we have with RGX-314. On the left are the subretinal delivery studies that we have completed in the case of the phase 1/2A study, and the first of 2 pivotal studies, Atmosphere, for treatment of wet AMD. And on the right are the 2 ongoing studies evaluating suprachoroidal delivery of RGX-314 for the treatment of wet AMD and advanced diabetic retinopathy without center-involved DME, respectively. So I'll first walk you through our more advanced program, our subretinal program. This slide shows the basic design of our completed phase 1/2 study. These full 2-year results were presented recently at your Retina for the first time by Dante Paramici. So in this study, we dosed 42 patients across 5 ascending dose cohorts. We followed the patient in a typical fashion with monthly visits up to the end of the 2-year follow-up. And once patients completed the 2-year follow-up, they extended into a long-term follow-up. We saw excellent safety and tolerability, and also saw what we were targeting to see, which was stable to improved visual acuity and central retinal thickness in the setting of a meaningful reduction in anti-VEGF treatment burden. And we saw this across the 3 highest dose cohorts, 3, 4, and 5. And in our 3rd cohort, which now has gone beyond 3 years, we've seen these benefits have extended out to that duration of follow-up, and we continue to follow these patients. So based on these findings, we advanced into pivotal development. First, the first of 2 pivotal studies, the Atmosphere study. The design is shown here. We started this study at the beginning of the year, and we're excited with the plan to proceed to start of the second of the 2 pivotal studies by the end of this year. Suprachoroidal delivery, we're very excited about these first ever studies evaluating gene therapy via the suprachoroidal space in the clinic. The first of these studies is the Aviate study evaluating previously treated wet AMD patients. Here's the basic design of this study, in which patients do not receive prophylactic steroids. We've completed dosing of the first cohort and also completed dosing of a second dose level in both neutralizing antibody negative and also neutralizing antibody positive patients. We've seen good tolerability. And last week, Nick London presented for the first time the initial results that we see from cohort 1 in this study out to at least 6 months, where we see stable anatomy and function, and again, a meaningful reduction in treatment burden. So we're very excited to advance in that study, and also the Altitude study, which is evaluating suprachoroidal delivery of RGX-314 in patients with advanced DR. And we've completed the first cohort, and we're currently enrolling in the second and third cohort. And I hope to see all of you on Saturday when Dennis Marcus will present for the first time initial results from cohort 1 at ASRS. Thank you.